Transcription is synthesis of RNA using DNA as a template. Here in this lecture today we are going to discuss how this transcription takes place in prokaryotes, and then we will discuss transcription in case of eukaryotes, especially with reference to the messenger RNA transcription. Different types of RNA molecules. The most important one with reference to gene expression is messenger RNA, which has a message for synthesis of proteins used on ribosomes. Non-coding RNA include transfer RNA, which transfers specific amino acids to the growing polypeptide chains during translation. And ribosomal RNA is a component of ribosome, actually, which is the place where this translation takes place. Then there are microRNA, which regulate gene activity by gene silencing and or gene activation in some cases. And then there are ribozymes or catalytic RNA, which have very important part in, especially in the evolutionary pathway. And these are the RNA which act as enzymes. This is the general overview picture of the transcription. Transcription takes place with the help of RNA polymerases. And here in the figure you can see this blue one is the DNA molecule, which is double-stranded structure, and this green is the RNA molecule. And this RNA is synthesized in the 5' to 3' direction. And at a given time, there is a small RNA DNA hybrid of about 8 base pair that exists, and this is called as the transcription bubble. And this transcription bubble continuously it goes towards 5' to 3' synthesis of RNA molecules, and this is the direction of transcription. And DNA before and after is rewound or unwinding of the DNA that takes place with the movement of this transcription bubble. This is mechanism of action of RNA polymerases. They require 3 aspartates and 2 magnesium as cofactor, and any incoming nucleotide is in the form of triphosphate, in which this monophosphate is added to the 3' end of the growing RNA chain. Here no primer is required. RNA polymerase can initiate its own chain. And as you can see here, there is a phosphodiester linkage is created by transfer of electrons to this phosphate, and this pyrophosphate is released. And this energy which is released out of this pyrophosphate is used by the RNA polymerase to move on the template. Some of the basic or fundamental terminology which is important for transcription. There are 2 strands of DNA. One is template strand and other one is the non-template strand. So which one is the coding strand here? As you can see here, this template strand is the one which is taking part in the reaction, meaning RNA is being synthesized by using this template strand as a template. And non-template strand is not participating here. But this non-template strand is called as coding strand because this non-template strand is exact copy of the RNA transcript. And here you can see only difference is of the U's. Uracil is present in RNA and thymine in DNA. So DNA template strand is the non-coding strand and non-template strand is the coding strand. Then other terminology which is important is the transcription is initiated at the promoter region. Promoter is a sequence of DNA present on the DNA molecule at a specific place from where transcription may be initiated. And every gene or every RNA molecule has its own promoter, and this from where this transcription is started, that is +1, and all the sequences towards right side are called as downstream sequences and labeled with +1, +2, and so on. And all the sequences towards leftward here in this case, they are designated as -1, -2, and so on. So there is no zero here, +1 and -1. And these are downstreams and these are the upstream sequences. Now here you are looking at basically the promoter region in case of prokaryotes. The prokaryotic cells have this -10 region and -35 region upstream of their uh, initiating part of the gene. So this -10 has a certain sequence, as you can see here, and -35 is here. And these are the consensus sequences. Consensus sequences are those sequences which are average sequences of many other sequences. As you can see below, there are uh, promoter regions of several genes in E. coli like TRP, LAC, RK, ARA, BAD, and so on. And these are their actual sequences, and these sequences here are the consensus sequences. And you might have observed that this -10 may not be exactly -10, but it is rounded to -10, meaning it is a -10 region, and this is -35 region. And in some cases in prokaryotes there is also UP element far upstream, as you can see in this gene over here, but these genes don't have this UP element, so it means UP element is present in some of the genes but not in all the genes. And this RNA transcription can go in any direction, like it depends on the sequence of the promoter, that in which direction it is present. Like here, this top strand is coding strand, and for many of the genes which are being transcribed towards right side, it is also by convention that the coding strand is written as the uh, strand for the name of for the sequence of the gene. And in this case, there may be certain genes which are transcribed in this direction. So their coding strand is bottom strand is the coding strand, meaning the non-template strand for these is this, and top one is the template strand, the genes which are transcribing in this direction. And genes which are transcribing in this direction, they have uh, this non-template strand is written over here and template strand is written over here. And in case of RNA transcription, in case of prokaryotes, only one type of RNA polymerase can form all three types of RNA molecules: messenger RNA, ribosomal RNA, and transfer RNA. And this you can see -10 and -35. This is the promoter region, and this is the RNA polymerase, which binds to this promoter region. And there is a closed complex which is converted into an open complex. Transcription begins and it goes on. So there are three phases: initiation, elongation, and termination. And here important factor is the sigma factor, which I will be explaining a little shorter, that this sigma factor, how it is important for transcription in case of prokaryotes. This is RNA polymerase uh, showing these different subunits, like 2 alpha subunits, beta, beta prime, this is sigma subunit, and omega subunit. So remember, as I mentioned, only one type of RNA polymerase is required for synthesis of all three types of RNA molecules in case of prokaryotes. And this is the overall picture of starting of the transcription. initiation of the transcription, and then elongation and termination. It is more clear from here. You can see there is, if for example there is promoter X, then there is a sigma X. So the function of the sigma factor is loading of the RNA polymerase on the promoter region, correct loading of the RNA polymerase. So here in this case, this sigma factor loads the RNA polymerase. So there is a closed complex and open complex. So there is green is the RNA which is being synthesized. After synthesis of a few nucleotides, the sigma factor is no longer needed and is released. And for elongation factors, especially this NUSA is the important elongation factor. This comes in place and it helps RNA polymerase to move and to make this RNA molecule. This is elongation phase. Finally, it is terminated when this some special sort of terminator sequence comes. Whole things falls apart. NUSA is released, completed messenger RNA is released, and there is a dissociation of polymerase, and now it is available for next round of transcription. Here hypothetical sigma y is shown here, so it means either sigma x or sigma y depending on which genes are going to be transcribed. That kind of sigma factor can bind to the RNA polymerase and initiate further rounds of transcription. So here you can see there are different sigma factors, these sigma subunits. Sigma 70 is the molecular mass. So this is, all these are shown with the help of their molecular mass, sigma 50, 438 and so on. And you can see major sigma subunit is the sigma 70, 700 molecules per cell. And these are meant for housekeeping genes. So most of the housekeeping genes, they are transcribed with the help of sigma 70. And other genes such as modulation of cellular nitrogen levels, sigma 54, and similarly heat shock genes, for example, sigma 32. So different sigma factors, they actually help initiation in the transcription of genes which are required according to the need of the cell. So if in case of there is a heat shock is given to the E. coli cells, certain sets of genes are activated or start transcription, and they are needed, though certain special proteins are needed. So in that case, this sigma 32 is activated and it binds to the RNA polymerase and loads on the genes which are required for heat shock genes. Similarly, for other types of genes. And how this transcription termination takes place in E. coli. There are two pathways. One is the Rho dependent. Rho, RHO. This is a protein which helps in the termination of transcription in E. coli in some of the genes. This Rho protein has a sequence specificity on the messenger RNA in some genes in case of prokaryotes, and this Rho protein, it runs along with this transcription when this transcription is going on towards the RNA polymerase, and then there is some sort of bumpy sequence comes. You can say there is some speed breaker comes, a sequence comes which slows down or pauses the RNA polymerase so that this protein catches the RNA polymerase and whole things falls apart. In this way, transcription terminated by Rho dependent, Rho protein. And in many other cases, there is no requirement of Rho protein, but those are called as row independent termination of transcription. In that case, there is a strong hairpin structure, as you can see here. This sequence is present in the DNA on the 3' end of the RNA. This sequence is present within the DNA molecule. When this sequence is transcribed into the RNA molecule, there is a strong intra-chain hydrogen bonding due to the complementarity between the chain. So, with this hairpin structure, it pulls apart the RNA polymerase and whole things falls apart and termination takes place. So it means row independent termination of transcription takes place in this way. Now coming to eukaryotic RNA polymerases, there are 3 different types of RNA polymerases: RNA polymerase 1, 2 and 3. RNA polymerase 1 is for ribosomal RNA except 5S ribosomal RNA. RNA polymerase 2 for messenger RNA and most of the small nuclear RNA and microRNAs. And RNA polymerase 3 for transfer RNA and 5S ribosomal RNA and some other small RNA molecules. So 3 different types of RNA polymerases as opposed to only one type of RNA polymerase in prokaryotes. As I mentioned earlier that I will be discussing more detail about RNA polymerase 2 and messenger RNA transcription in eukaryotes. Although transcription of transfer RNA and ribosomal RNA is also very important, that protein cannot be made without if we miss any of those. But messenger RNA is the most important with reference to the gene expression because it carries the message. And you may say that transcription in case of ribosomal RNA and transfer RNA is also very much similar as far as the RNA polymerase mechanism is concerned, and there are certain promoter regions are there for them as well. But there is more regulation takes place in case of RNA polymerase 2 because it depends which genes are going to be expressed in which situation. So here is the promoter sequence. In this case, there is a sequence which is called as Tata box. It is present in -30 region. Then there are various regulatory sequences present far upstream, and in some cases downstream even there are sequences which are present and which help in the regulation of this transcription. But this sequence is most important at -30 TATAA sequence, and this is the place for first nucleotide that comes in case of messenger RNA in eukaryotic cells. And there are multiple factors which are required for before this RNA polymerase can bind to this promoter region. The first protein is TBP, Tata box binding protein, and this protein along with factor TF2A. So you can see here the factors. TF2 means transcription factor for RNA polymerase 2. Similarly, for ribosomal RNA, there will be TF1, and for transfer RNA, there will be TF3, depending on RNA polymerase 1 and 3. Here, transcription factor 2A, and then there is a transcription factor which is 2B. After the binding of these two, then comes the polymerase 2 along with transcription factor 2F. Here you can see 2F, and then transcription factor 2E and H. They bind here, and this whole complex is assembled, and this is called as closed complex. Here, very important is the CTD, carboxy terminal domain. We will see it has great significance in RNA polymerase 2 transcription in eukaryotes. carboxy terminal domain or CTD of the RNA polymerase. Now this TF2H performs two very important functions. First of all, it acts as an helicase and opens the duplex, and this is called as open complex. And second important function is this also acts as a kinase, and it phosphorylates this CTD of the RNA polymerase 2. With this phosphorylation, this transcription is initiated, and some of the factors remain behind, and later on TF2H and E, those are also released, and this uh, goes on the DNA template in the 5' to 3' direction. Then elongation factors come in, they bind, which help in this transcription. And finally, there is a termination takes place. I will discuss there is a special type of requirements and proteins for termination of transcription in eukaryotic cells that I will be discussing a little later. And finally, whole things falls apart and transcription is terminated in this case. And this is just the RNA polymerase 2 showing how this pocket is created and from where this RNA exit that takes place, and there is a transcribed DNA upstream and entering DNA downstream. So RNA polymerase is a big protein, multi-subunit proteins, and DNA, how it is encountered, how this RNA is generated, and you know magnesium is required for its function of RNA polymerase. And here you can see different proteins. First is the RNA polymerase 2 itself with 12 subunits. Then tata binding protein is just one subunit, and then TF2H, and these have, and 2H is also 12 subunits. Their molecular masses are given there and functions are given here. And you can see the most important functions, TF2H, unwinding and kinase function of this, and others also have certain functions. And this in elongation, there are certain factors which are required for elongation of the RNA polymerase 2 transcription, and there are these are some of these are single subunits and other are the multi subunit proteins. Now this transcription when it's taking place after a particular small part of the RNA is made, there are certain changes that come in the RNA molecule in eukaryotes. These are called as post-transcriptional changes, and these post-transcriptional changes does not mean that transcription is totally completed and then these changes occur, but these occur simultaneously while still transcription is going on. So three most important one are, number one is the addition of a 5' cap. This cap is a special sequence, as I will be discussing. This has important functions. One is it brings stability to the messenger RNA. Without this cap, nucleases attack on the RNA messenger RNA and degrades it. Second is it also has a role in the translation. Then you might know about introns. These are the sequences which are present within the genes, but these are non-coding part, meaning these are removed from the mature messenger RNA. So these introns, they are removed by a process called as splicing. So this splicing takes place while this transcription is continuously going on. And then the last one is the cleavage and polyadenylation. Cleavage means breakdown, and polyadenylation means addition of a poly-A tail, adenosine residues at the 3' end. And these several A's, about 200 to 250, they have special functions. First, they also gives stability to the messenger RNA at the 3' end, then they help in the transport of messenger RNA from nucleus to the cytoplasm. And finally, they also have a role in translation. So these three important post-transcriptional changes of messenger RNA in eukaryotic cell, I will be discussing these in detail one by one. The first is the 5' cap. This is the sequence of the cap 7-methyl guanosine. It means there is in the guanine at the 7th position this methyl group is there, CH3 is there, and there is an unusual linkage of triphosphate linkage from first nucleotide in the messenger RNA, and this extra sequence is added here. 5' triphosphate linkage is there. And then in some cases, sometimes this first ribose is 2' hydroxyl is methylated, and in some cases the second nucleotide, ribose of the second nucleotide, is also methylated at the 2' hydroxyl position. So this whole structure, as I mentioned, gives stability to the messenger RNA, and this also has a certain special role in translation. And this is enzymatic process. There are certain enzymes which is quite simple. You will see to some people these may look quite complicated, but actually they are very simple. So first thing is, this is the growing 5' end of the RNA. When about 30 nucleotides of messenger RNAs are made, the RNA comes out of the RNA polymerase, and this 5' end, it contains, the first nucleotide always contains 3 phosphates at its 5' end, and all the remaining they have monophosphate, because triphosphate comes, as I mentioned, in the reaction of this RNA polymerase, and monophosphate is added, pyrophosphate is released. But the first one has 3 phosphates, alpha, beta, and gamma. So first there is a hydrolysis takes place of the phosphate. So phosphohydrolase is the enzyme. This gamma phosphate is released by this reaction. So you get 2 phosphates now. In the next step, there is an addition of guanylyl transferase is the enzyme, and addition of a GMP. So guanine monophosphate is added. So GTP comes in, pyrophosphate is released, and this GP is added here. So this in this case there is a 5' triphosphate linkage, and this guanosine is added to this to these. So these 2 phosphates are these 2, and this one is from the GMP. Now this position, the 7th position, is methylated, and again very obvious enzyme, guanine 7-methyl transferase. It transfers methyl group from methionine to homocysteine, and this is synthesized like 7-methyl guanosine. And as I mentioned, in some cases this position is also methylated. So here 2' O methyl transferase. So it means methyl group is transferred at 2' O group here. So here and here with this enzyme, and this cap is completed. So you can see it is not as much complicated as it looked in the beginning. So there is a sequential way of adding or releasing. For example, first phosphate is released, GMP is added, methyl group is added here, and then here. So in this way this cap is synthesized. Now, as I mentioned earlier, that this CTD has a very important role. So all these enzymes, they do not come from the medium, but they are already sitting on the CTD of the RNA polymerase and moving along with the RNA polymerase too. And these are called as cap synthesizing complex. All these enzymes phosphohydrolase, methyltransferase, guanylyl transferase, and they are sitting over there. So as soon as 5' sequence comes out, these enzymes start function and they add this 7-methylguanosine and other methyl groups. And once this cap is generated, these enzymes are no longer needed, so they are detached from this CTD. And now comes other proteins which are called as cap binding complex proteins, shown here in blue. So these proteins bind this cap to the CTD. So this 5' end of the messenger RNA is not being held free but in the nucleus, but it is in the bound form in this way, and this transcription continuously it goes on. Now second post-transcriptional change, which is splicing. This is overview, like introns are there which are released and exons, they are joined together. Splicing, there are different types of splicing and they are categorized in group 1, group 2, class 3 and class 4. Group 1 and group 2 type of splicing mechanisms, or group 1 or group 2 introns, they are quite interesting in that way that they also act as enzyme, meaning they do not need any protein. They can perform function by themselves. So I first explain a little bit about this splicing in group 1 and group 2. Group 1 is found in bacteriophages as well, messenger RNA, transfer RNA, ribosomal RNA of lower eukaryotes, and prevalently in higher plants, and also in transfer RNA and ribosomal RNA in case of bacteria. So I want to make it clear here is that the introns in messenger RNA are only present in eukaryotic cells, but in transfer RNA and ribosomal RNA, these may also present in bacterial cells as well. And how this reaction takes place? There is an attack of a guanosine from outside to this part. So this green are the exons, and this yellow part is the intron. So this hydroxyl attacks here, and it attaches itself, this guanosine, to this adenosines, and this hydroxyl becomes free at the uracil. The electron pair attacks to this end, and these exons are joined together, and this intron is released. So this is group 1 type of splicing, and in case of group 2, it is found in mobile genetic elements in bacteria, organelle elements of archaea even, and in eukaryotes. So Thomas Cech is the person who discovered this very important part, that RNA can act as enzyme, and you may know that there was a big question whether DNA comes first or protein comes first, because without DNA, protein cannot be synthesized, and without proteins, DNA cannot be replicated. So this question, people have been asking this question from centuries. Ultimately, answer came from the RNA, that it is neither DNA nor protein. It is RNA which perform this function, because it does not require any protein. So it can make itself without using any protein, and it also can act as enzyme. Not only in splicing, we will see it can add nucleotides to itself as well, which has very important implications in evolution. So here in group 2 type of intron splicing, here even there is no need of nucleotide from outside. So here this adenosine itself attacks to this part, and it releases this nucleotide from here, and this laureate structure is formed. And this hydroxyl, as in the previous group 1 intron splicing, it attacks to this end, and this axon, these two axons are joined together or spliced together. And this laureate structure is released as intron. Now coming to the most important one, as we were discussing about messenger RNA, how this splicing takes place in messenger RNA. In case of messenger RNA, there are 5' axon and 3' axon for each splicing step to take place, and inside is the intron. And here these are not self-splicing, but they require proteins, and these proteins contain RNA in them. So these are called as U1, U2, and few more. And these proteins contain RNA, and these RNA contain some special nucleotides like phi in this case. They have this complementary sequence to this axon intron junction, and within the intron, somewhere within the intron. So these proteins recognize this axon intron junction phi prime and also recognize this, and then how they perform it, I will show you here. And these protein and RNA complexes, these are called as SNRPs, SNRNPs, small nuclear ribonuclear proteins, and they bind to these axon intron junctions and follow this step of splicing as explained in the next. It is more clear from here that first of all U1 SNRNP and U2 SNRNP, they bind to this region U1 and U2 within the intron, and this is ATP dependent process. ATP is hydrolyzed, and then a complex of U4 and U6 that binds, and U5 also comes in place. So this is called as inactive spliceosome. Another ATP is utilized here. This inactive spliceosome then is converted into active spliceosome by removing this U1 and U4, and another ATP is utilized. So it means this is a tightly regulated process. It requires energy, and this is an active spliceosome now. Where the similar process as in group 2 intron splicing took place, that this axon is intronic part that goes this G, it goes to the U, and this laureate structure is formed. And then finally these axons are joined together, and this intron in the form of laureate structure is released. So in this way, this splicing of messenger RNA takes place in eukaryotic cells. And once again, the interesting part is that this spliceosome machinery is also present on the CTD of the RNA polymerase 2. This CTD has this SNRPs, and as soon as intron comes, these SNRPs process that intron by the procedure I just mentioned, and this spliced intron is removed and these axons are joined together. So meaning this CTD also has important function in splicing as well. Now coming to the last part of this post-transcriptional changes, 3' end processing or cleavage and polyadenylation. Now how this termination takes place, transcription termination in eukaryotic cells in messenger RNA. there is a conserved sequence of AAUAAA, and this sequence of course is present in DNA as AATAAA, transcribed into this RNA. And there are certain proteins that bind to this hexamer sequence, and this is hexamer because of these 6 nucleotides, and they release the messenger RNA and polyadenylate the messenger RNA from on the 3' end. And here energy is utilized once again, and the major enzyme which perform this function of polyadenylation is polyadenylate polymerase, but there are other proteins that I will show you. But remember, this cap is not like that. Actually it is like this. So this cap is bound here, and hexamer, when it comes here, the machinery for this cleavage and polyadenylation is also sitting on the CTD, carboxy terminal domain. And this is CTD, and this is the machinery. These are the proteins. So here some of the proteins are shown, like CPSF. They have 4 subunits, CSTF having 3 subunits, polymerase, cleavage factor 1 and 2. So, and the 2 sequences which are important, one is the hexamer highly conserved sequence AAUAA, and other one is the GU-rich or U-rich or combination. GU-rich means some of the GU sequence, and U-rich means some of the U's are there or their combination in. So these is variable in different genes. So these proteins, CSTF binds to this region, CPSF binds to this region. These blue 4 subunits are of CPSF. And this is polymerase, and these are the cleavage factors. And this CPSF 73 cleaves this RNA from here. And this polymerase, it adds the adenosine residues 200 to 250 long. This tail is formed, and this termination of transcription also takes place here. The whole thing falls apart, and messenger RNA goes out of the nucleus into the cytoplasm for translation to take place. Here is an example of a gene, oval albumin gene, 7700 base pair long gene with all of its 7 introns, and it is transcribed into messenger RNA. 5' cap is added, and on the 3' end poly A tail is added, and you can see these 7 introns are spliced out by splicing process, and final mature messenger RNA is only 1872 nucleotide long. So a couple of things here. In case of eukaryotic genes, they contain sometimes just a few, 1 or 2 or few introns, and sometimes even 20, 25, many introns. Only a few genes, they do not contain intron at all, like histone genes do not have introns. And another important thing is intron sizes are much larger as compared to exons. As you can see here, the oval albumin gene is 7700 base pair, whereas processed mature messenger RNA is only 1872 nucleotides. Some introns are like 50 to 60 kb long. The gene size is along with introns, but their mature messenger RNA is only 5 to 6000 base pair, nucleotides, 5 to 6000 nucleotides long. So introns are much larger. Alternative cleavage and polyadenylation may make different proteins from the same messenger RNA. Like there is only one gene and there are 2 polyacids, A1 and A2, and these 2 sites, it depends which one is processed. If A1 is processed, shorter messenger RNA and shorter protein will be made. If A2 is processed, relatively longer messenger RNA, hence longer protein will be made. It means from one gene you can have two different proteins. Similarly, in alternative splicing, if a splice site is missed, is not used, you will find you will have different size of messenger RNA, hence different protein. As in this case, you can see if it depends which splice site is used. So in this case, there is longer mature messenger RNA, and this is shorter messenger RNA just because of difference in splicing which is used. So this is a very good example of alternative processing. This is a primary transcript having these introns and exons, and here there is one polyacide here and another one is here. You know, DNA and genes are same in all types of cells within an organism. What is the difference is the expression. So here this is the same gene. When it is expressed in thyroid, this polyacide is used and you get this size messenger RNA and you get calcitonin. Whereas in brain cells from the same gene, this polyacide is used and this is not used. Now how this is regulated? Of course, this is not purview of this lecture. I have discussed this in another lecture in gene regulation and expression. But here I just discussed that if this polyacide is not used and this one is used by some mechanism, so in the brain cell, this is what happens. And you get a CGRP, which is shorter protein. So it means there is a conservation of the genes. You can say the same gene just by alternative polyadenylation or by alternative splicing give us different proteins. And these are splicing patterns that can take place. There is alternative 5' splice sites or alternative 3' splice sites, and different examples how this alternative splicing may produce different sized messenger RNA, hence different proteins. In case of RNA polymerase 1, it makes ribosomal RNA except 5S ribosomal RNA, and here class 1 transcription factors are used, TF1, and in polymerase 3, TF3 are used, as I mentioned earlier, but I am not going in detail of these here. And there are some modified bases which are used in ribosomal RNA and transfer RNA. We will see, especially in case of transfer RNA, we will see different modified bases. They are produced by post-transcriptional reactions, and these are some individual post-transcriptional reactions which alternate or modify these nitrogenous bases. Now the processing of pre-ribosomal RNA transcripts in prokaryotes as well as in eukaryotes. First I will discuss this pre-ribosomal RNA transcript processing in case of bacteria or prokaryotes. Here we have pre-ribosomal RNA transcript of 30S size. S is the Swedberg constant. There was a weird value of sedimentation coefficient that when multiplied by some factor, you get a reasonable value number like 30S. So it depends on basically is ultracentrifugation of these components, how they behave like, and what is their sedimentation coefficient, and that is converted into Swedberg constant after the name of the scientist. And as you know, one type of RNA polymerase can form all types of RNA transcripts in bacteria. So this pre-ribosomal RNA contains 16S, 23S and 5S ribosomal RNA and transfer RNAs. In the processing, there is methylation takes place, and pseudouridine or dihydrouridine conversion also takes place, modification of the nitrogenous bases. This methylation protects this RNA from cleavage by nucleases. Here, as opposed to splicing, there is a cleavage, meaning when it is broken down into different components, while in splicing, the exons are joined together. Here no joining takes place, and this cleavage takes place in two steps, like these are the intermediates, and finally you get the processed like 16S, 23S and 5S ribosomal RNA and transfer RNA. In case of eukaryotes, this is an example of ribosomal RNA processing, and here there is a much larger size 45S ribosomal RNA, and remember this takes place within the nucleolus of the nucleus. The genes for ribosomal RNA traversed through the or pass through the nucleolus inside the nucleus, and ribosome is also assembled within the nucleolus, and then they come out of the nucleus. And here this is pre-ribosome of 90S, and these are the three ribosomal RNA components. As you know, the 5S is not made by RNA polymerase 1, but with the RNA polymerase 3. So RNA polymerase 1 formed this pre-ribosomal RNA transcript in eukaryotes, and small nucleolar RNPs, they actually perform this function, and there is similarly methylation, pseudouridine and conversion into the other modified nitrogenous bases, and initial cleavage forms these ribosomal RNA intermediates, and here 5S ribosomal RNA also comes in the picture. From it is synthesized from RNA polymerase 3, and whole of this is assembled. So this is a pre-40S and then the pre-60S ribosome containing the ribosomal RNA, and finally there is further cleavage and then export into the cytoplasm takes place. So mature ribosomal subunits, they contain 40S ribosome having 18S ribosomal RNA, and this 60S ribosome containing 5.8S, 28S and 5S ribosomal RNA. So in this way, processing of pre-ribosomal RNA transcripts in eukaryotes takes place. And in case of transfer RNA, the processing takes place both in bacteria and eukaryotes, and there is enzymatic breakdown like RNase P cut, RNase D cut, so they cut these nucleotides. Then there is an addition of CCA at the 3' end, both in case of prokaryotes and eukaryotes, and there are modification in the bases that also occurs here. And in eukaryotes, there is a splicing that takes place of the intron. This intron is removed and exons, they join together. Another type of RNA is microRNA. It is synthesized with the help of RNA polymerase 2 in the nucleus by eukaryotes, and there is longer primary microRNAs are synthesized as here, and you can say there is a lot of intra-chain hydrogen bonding. And this enzyme, the DROSHA enzyme, it converts this primary microRNA to pre-microRNA by cutting from various places, and it is exported out from the nucleus to the cytoplasm. Here dicer enzyme acts and dicing takes place, meaning this single-stranded intra-chain RNA molecule is converted into double-stranded molecule by cleavage on one side, and then this RNA helicase that unwinds this double-stranded molecule into single-stranded ones, and then there is RNA-induced silencing complex, certain proteins. This RNA is loaded on this RISC. Now, what is the function of this microRNA? One major function is gene silencing, which is shown here, and there are two pathways or two mechanisms. In one case, the ultimate objective is to silence the gene by hindering the messenger RNA. So in one case, there is a near perfect complementarity to the messenger RNA which is produced with this microRNA. In that case, what happens is mRNA is cleaved by nucleases when this duplex is formed. In another case, there is a partial complementarity, and in that case, there is a translational repression. Messenger RNA cannot be translated, although there is no degradation. But in both cases, this messenger RNA that is silenced by this microRNA because it is complementary to the messenger RNA. This is a secondary structure of self-splicing ribosomal RNA intron of tetrahymena. Here there is a long intron that are having intra-chain hydrogen bonding, and this is the exonic part shown by red and blue arrows. These are shown showing the splice sites, and this part is the catalytic core of self-splicing activity, this shaded part. So basically this is an example of ribozyme. And this in vitro catalytic activity of L19IVS, here in this case this L19IVS that is generated by autocatalytic removal of the 19 nucleotides from 5' end of the spliced intron, and the cleavage site is indicated by an arrow. This is the cleavage site in the internal guide sequence. This is the guide sequence in the box. The G residue added in the first step of the splicing reaction is part of the removed sequence, and a portion of the internal guide sequence remains at the 5' end of L19IVS. So this is a good example of in vitro catalytic activity of RNA. And here this shows that this RNA can act as an enzyme by extending its chain by itself. This is, although this is in vitro activity, but what happens here is that there are 5 C's here, and this is an intron, and these 5 C's, out of these 5 C's, one is attached with this G, and these 4 are left. Then 5 more C's comes in, like a chain of 5 cytosines. They comes in, and once again this time what happens? The opposite of this reaction takes place, and this chain of 5 takes out this cytosine, and it becomes C6, 6 cytosines here, and this remains as such for another round. So this is a very significant reaction evolutionary point of view, which shows that if you just need a couple of nucleotides, they can add by themselves and make a longer chain. And once you get the longer chain, further translation, there is a chance of translation that takes place, and further life can originate from that. So it means if you have few molecules of inorganic converted into some organic molecule by Miller's reaction, and those organic, if those have these nucleotides, they can add themselves so that life can be originated. This is one view of evolution. This is central dogma that DNA can be synthesized from DNA, and DNA to RNA, and RNA to protein. But there is a change in the central dogma that RNA can be converted back into DNA, or RNA can be replicated by itself. Here is one example of HIV, which is a retrovirus. Its RNA genome, this virus, when attacks its host, it goes, this RNA comes in, converted into cDNA by reverse transcriptase reaction, and it may be integrated into the chromosome, and whenever required, it may come out and starts functioning. So this is an example of a reverse transcription, and this reverse transcription is also present in eukaryotic systems as well. Like I gave you an example in my replication lecture about the telomeres and telomerases. This telomerase is also a type of reverse transcriptase reaction. And this is an example of SARS-CoV virus and MERS, related to the coronavirus. These are also RNA viruses. So they have a little different function as compared to HIV. Their coat, when it is attached to the receptor on the cells, this virus comes inside the cell. Once inside the cells, there are certain, because they have a single stranded positive strand RNA, so that RNA is directly translated and makes some proteins, including a protease. And this proteolytic action by the protease makes certain enzymes, especially replicase, which helps in the formation of RNA, further RNA once again. And this is a negative strand RNA, and then there is a further transcription generating different sizes of RNA molecules, which makes different proteins which are required. And these proteins are made on the endoplasmic reticulum, rough endoplasmic reticulum, as we will see in our lecture in translation, that this proteins which have to be exported out, they are made on the ribosomes present on the endoplasmic reticulum. That's why it is called as rough endoplasmic reticulum. And there is a post-translational changes that take place here as well. And finally, there is an assembly of this virus that takes place, and positive strand RNA is also generated here with the RNA replication, and finally this virus goes out of the cell to infect the neighboring cells. So this is another example of RNA. Now these cellular messenger RNAs have different degradation rates. This degradation rate is also important to consider, because concentration of any molecule depends on its rate of synthesis as well as its rate of degradation. So nothing is like permanent in the cell. Rather, these RNAs are made for a very shorter period of time. For example, average half-life of messenger RNA of vertebrate cell is about 3 hours. Although there are certain RNA molecules that can last longer, but this is an average half-life. And in case of bacterial messenger RNA, only 1.5 minutes, perhaps because of regulatory requirements. So half-life is very small. It means there is a continuous process of synthesis and degradation of these RNA molecules taking place. Now, how is this messenger RNA degraded by ribonucleases in different cells? In E. coli, the process begins with endoribonucleases, which cleave or make several cuts within the RNA molecule, followed by 3' to 5' degradation by exoribonucleases. Whereas in lower eukaryotes, the major pathway involves shortening of poly-A tail and decapping of 5' end, and then degrading the messenger RNA in the 5' to 3' direction. In case of higher eukaryotes, 3' to 5' degradative pathway also exists. And all eukaryotes have complex of up to 10 conserved 3' to 5' exoribonucleases, and these are called exosomes. So it is to remember that 5' cap and 3' poly-A tail are first have to be removed, and then further degradation can take place in eukaryotes. Polynucleotide phosphorylase is an enzyme which makes random RNA-like polymers, and this is template independent reaction. It's not like RNA polymerases, so it is non-specific. So it can just extend the chain non-specifically if needed. With this, I end my lecture on transcription, and for gene regulation and expression, you may find my other lectures where I have discussed how these genes are regulated and expressed in different conditions. Thank you very much.